Hello everyone, my name is DAYANG. Today I will show you how the concept of rotational motion is applied in maths with computer graphics to create 3D animation by using C. Let's dive into the code and its output. Okay, so this code creates a spinning 3D cube using principle of rotational motion. First, we define the cube vertices in 3D space. Next, we apply rotation matrices to calculate how each vertex moves around the x, y, and z axis. And finally, last but not least, we project the rotated points onto a 2D screen using symbols like alias and asterisk to display the cube. Okay, so here's the output. A 3D cube spinning dynamically. Each frame updates the cube's position by applying new angles for rotation. These continuous transformations give us the illusion of motion. The combination of rotation matrices and projection is what makes 3D animation possible in programming. Okay, now in summary, this code illustrates how rotational motion and mathematics bring static objects to life in 3D animation. Thank you.
PCR, or polymerase chain reaction, is a widely used method in biotechnology to amplify DNA samples. A thermocycler, as shown in the slide, heats up water to evenly heat up the samples to the required temperatures without overheating the samples. This is because hot water transfers heat energy to the samples via conduction. And due to the high specific heat of water, this thermocycler can transfer heat energy without overheating each sample. During denaturing step, the thermocycler heats up the samples till 90 degrees Celsius. This helps the DNA, double-stranded DNA, to unwind. During annealing step, the sample is cooled down to 60 degrees Celsius. This allows the primer to bind to the single-strand DNA. Finally, during extension, the sample temperature is raised to 72 degrees Celsius for extension by the Taq polymerase enzyme. These three steps are repeated multiple times to allow for multiple DNA amplifications.
On to thermodynamics. This is the first law of thermodynamics, stating heat energy cannot be destroyed or created, but can be transferred either from one location to another and converted to or from other forms of energy. Exercising, brain activity, muscle growth. All these activities require energy. The energy is in the form of ATP, which is the energy currency of cells. ATP is used to break down glucose into pyruvate and then sent to other reactions like ETC and Lip reaction to make more of the ATP for energy usage. As glucose is hydrolyzed into pyruvate, all the bonds of glucose is broken down and conserved as energy in the form of ATP to be used by the body.
Hi everyone, now I will briefly talk about the second law of thermodynamics, focusing on the entropy changes in reaction and diffusion. The second law of thermodynamics states that the total entropy of an isolated system always increases over time. Entropy is a measure of disorder or randomness in a system, where the system tends to evolve toward the maximum entropy when the energy is evenly distributed. For example, when baking soda and vinegar react with each other, in which the initially a stable and ordered compound, they will produce carbon dioxide, water, and sodium acetate. The release of the carbon dioxide, which is the bubbles, indicates that an increase in entropy. This principle is crucial in biotechnology processes like fermentation, where organisms like yeast release gases as they metabolize sugars, driving reactions that increase entropy. Now, let us see the entropy changes in diffusion processes. When a drop of food coloring is added into a water, the dye molecules are highly concentrated in one spot. Over time, they spread throughout the water due to increase in the system's entropy. This will happen until they reach equilibrium with the dye evenly distributed. This diffusion process is essential in biotechnology, specifically the use of liposomes to deliver drug to target cells. That is all from me. Thank you.